Okay, this is going to be DNA duplication. And first thing I'm going to do is just real quick draw my strand, my DNA double helix. All right. And the first thing we want to do is we need to open up the DNA so we can start duplicating. So in order to open up the DNA, we need helicase. Green will be helicase. Helicase is going to open up the strands as we move along the duplication process. Topoisomerase is going to be right next to the helicase. Use black. Topoisomerase basically just relieves the tension that the helicase causes by pulling apart the strands, right? Because if we pull them apart, it's going to create tension further down this end, or if we're going this way on this end. I'll draw it on both sides. Okay. So now we'll talk about SSBPs. So these lines right here, these will just be our bases. SSBPs, also known as single stranding binding proteins, they bind to the bases and prevent them from reattaching. So it's just another thing that helps keep the whole piece of DNA open. All right, now it's time to start duplicating. So as we've discussed before in class, we read DNA from 3 to 5 and make DNA from 5 to 3. All right, so for the sake of this video, we'll say that this end is the 3'. So that means this is 5'. Opposite at the bottom, so this would be 3'. This would be 5'. All right. So if our replication fork is right here, we're going to the right. We're going to be going from left to right and making the DNA. But the DNA is actually made from the 5 to 3'. And I'll kind of explain that in a second. First we need to do is we need to lay primer. So primase is going to come in. I'll use the black. Primase is going to come in and it's going to lay primer. This little black line right here, that's primer. All right, next is DNA polymerase. DNA polymerase is going to come in and actually start duplicating the DNA. The green, it's DNA polymerase. So we have our template strand right here in the red, and the green, the DNA polymerase, is making our complementary strand, right? So we're reading from 3 to 5, but we're actually making from 5 to 3, right? And our replication fork is going to the right. Now we need to discuss the bottom. So since, again, 3 to 5, 5 to 3, we make DNA 5 to 3. The issue is we're eventually going to run out of room. We're going to go this way, and we're going to out of room. So in order to keep the duplication process going at the bottom, we make what's called Okazaki fragments, right? And it's still just primer and DNA polymerase, but it's just in fragments. Like that. So it makes like these fragments called Okazaki fragments. So what happens is, so the green is our DNA polymerase. It's going to run into the primer. When this happens, it's just going to get rid of it so we can actually make a continuous strand at the bottom here. And in order to fuse those two fragments together, DNA ligase, we'll just draw like a circle right here. That'll be like the binding site. DNA ligase is going to fuse the DNA together at that point. And then the same thing will happen over here, right? So we're going to the right. DNA polymerase will come in, get rid of that primer. The DNA ligase will fuse them together. All right. So this would be our lagging strand because we have our leading strand at the top here leading into the replication fork going to the right. This would be our lagging strand. So I guess think of it as like one step behind. It's got a little bit more of a process that has to do with the Okazaki fragments, getting rid of the primer, and the ligase, the DNA ligase. And that's it. That's DNA duplication.